The topic for today's session is cloning vector. Now, first of all, let us understand what are vectors. Vectors are, when we talk about in biotechnological term, they are the DNA material which are capable of carrying any kind of foreign DNA which can be attached to itself. From where these vectors can be done, they can be found. You can have the whole list in the slide. I'm just reading the slide. Vectors are the DNA molecules that can carry a foreign DNA segment into the host cell. Now, vectors can be of three types which you can mention, or they can be more than that also. One of them is plasmids, which is one of the most commonly used vector in case of biotechnology. What are they? They are autonomously replicating circular extra chromosomal DNA. Another one can be bacteriophage. They are viruses infecting the bacteria. And the third one is cosmids, which are hybrid vectors which are derived from the plasmids, which contain cos site of lambda phase. Now, this plasmids we have been discussing in the previous sessions also. What is actually plasmids? In a prokaryote, especially in a bacteria is having a nucleoid. It is having its own DNA. Apart from that, there is a small extra chromosomal material present in the bacteria which is in a circular form. This is called as plasmid. And this plasmid has nothing to do with the nucleotide present in the bacteria. It has an ability to self-replicate. It can replicate by itself. It doesn't need any permission from the nucleoid of the bacteria to replicate. This particular property of the plasmid makes this plasmid really very useful in the bacteria. Plus, this plasmids, they have very less number of nitrogenous bases. They have a very less number of nucleotides. So, when you have to attach any other foreign DNA to it, you don't have to deal with the whole chunk of DNA of the cell. You just have to deal with few nucleotides which are present in the plasmid, and there you can attach your gene of interest in it. So, this plasmid, they act as vectors. Vectors meaning any kind of structures which are capable of transferring. They have an ability to carry something with them and transfer it to the other entity. If you did remember in Form 9, you have studied about vectors. What are vectors? Vectors are the carriers which carry pathogens from a sick person to a deceased person. So that is related to the virulence, that is related to the disease causing ability. But here in biotechnology, what are vectors? Vectors are DNA combinations. They are extracellular material which can which allow the foreign DNA to attach with them, and they can carry that foreign DNA attached to itself, and they can insert themselves into the host cell, so that that foreign DNA can also be inserted along with them into the host cell. So they carry the foreign DNA. So that is actually the function of plasmids. Bacteriophage, we all know what are bacteriophages. They are viruses which infect the bacteria. Now, when the virus infects the bacteria, you can see in the second diagram what it does. It sits on the bacteria, and then the viral DNA is actually injected inside the bacterial cell, and then the bacterial cell, the DNA of the bacterial cell incorporates the DNA of the virus, and then the DNA of the virus will also replicate along with the DNA of the bacteria. So, if by any kind of mechanism we can insert our DNA of interest into the viral DNA, so once the virus is going to infect, it will give its viral DNA inside. So, along with that viral DNA, our DNA also goes in. Our gene of interest also goes in. And once the viral DNA is replicated, along with that our gene of interest will also get replicated. So in a way, even bacteriophage, or it can be more specific, disarmed. Disarmed means they don't cause a disease. So first you treat these bacteriophages, and all the segments of the bacteriophages which are responsible for causing disease, you delete them out and then use that bacteriophage as a vector. Then it can be done in case of this recombinant technology. Third one is cosmids. Cosmids are something similar to the plasmids, but they will be having a site called as cos site with lambda phase in it for its proper function. So these are some types of vector, but the function remains the same, that they carry the foreign DNA with them into the host cell. Now, to be a good plasmid, to be a good vector, what are the properties a vector should have? Let's find out that thing. This PBr322 was the first plasmid which was used as a vector, and it was formed by two scientists. One is Bolivar and another one is Rodriguez. So that is why this, if you see in the name PBr, so P is the plasmid, B is the first letter of Bolivar and R is the first letter of Rodriguez. So this was the first vector which was constructed and which was used in the biotechnology as a vector, and this was a plasmid. So let's now what we are going to study is that we are going to study this PBr322, because this PBr322 is considered to be an ideal vector, and it contains all the properties, all the characteristics which a vector should have, which will make a vector proper tool of recombinant technology. So what properties it should have? These are some properties which you can see here. A vector, a cloning vector, should have origin of replication, should have selectable marker entities present in it, should have selectable marker for recombinant molecules, and should have cloning sites. We start with the first one, that is origin of replication. Here the diagram is of PBr322, an extended diagram of it which you can see in the slide. Now what is origin of replication? I am reading the slide. This DNA which is responsible for initiating the replication, any piece of DNA when linked to the sequence can replicate inside the host cell. ORI also controls the copy number of the linked DNA. Copy number is defined as the number of copies of vector present in a cell. It varies from 1 to 100 copies per cell. This origin of replication means a set of, you can see in the diagram, a set of genes which has this property, that how many times this particular plasmid is able to replicate when the plasmid will be inserted inside the host cell or on the nutrient medium. Just it gives an idea that how much copies this plasmid can make. So if the information is written fine in this origin of replication, that plasmid can replicate 2 times, so it will replicate twice. Or it is written, or it is there in it like it will replicate for 10 times, so 10 copies will be formed. Or it will be written that it is 100 copies, 100 times, then it will be 100 copies which are formed. So origin of replication tells how many copies the plasmid is going to make, plus it will have this origin of replication is the site from where the replication begins. So it is the initiating site in the whole plasmid from where the property of replicating will begin in the vector. Plus it will also govern the copy number of the vector. So how many times the vector will be replicated and from where the vector will be replicated is governed by this origin of replication, which is in short form called as ORI. There are other parts also in the plasmids. There are some selectable markers, there are cloning sites, which we are going to see in our next sessions. At this point, let us have a look at a video.
Any newcomer who joins a molecular biology lab will undoubtedly be asked to design, construct, or modify a plasmid. or billions of bases that make up the entire genome, plasmids typically contain a couple thousand at most. They are relatively small, stable, and easy to manipulate. So where do plasmids come from? In nature, they're found in microbes like bacteria. In the 1940s, scientists began noticing that there are heritable cytoplasmic factors that could be transferred between cells, but there was no consensus on what they were or what to call them. It wasn't until 1952 that Nobel laureate Joshua Lederberg coined the term plasmid, a combination of the words cytoplasm and id, Latin for it. In nature, plasmids often contain genes that provide a competitive advantage, giving its host bacterium an ability that it didn't have before. These benefits include antibiotic resistance, the wherewithal to survive in harsh environments, and even the ability to wage war to gain an environmental advantage. But plasmids aren't just useful to bacteria. Because they're incredibly easy to manipulate, they've also made themselves indispensable to life scientists and bioengineers. Plasmids created in the lab are known as constructs, or vectors. To understand what plasmids can do, let's break down its parts using a plasmid map. All plasmids contain an origin of replication, or ORI. It tells the plasmid where to begin replication. Plasmids often contain genes that are advantageous for survival. One of the most common, naturally occurring types of genes is antibiotic resistance. When used in the lab, antibiotic-resistant genes allow scientists to separate out cells that contain plasmids from those that don't. The wonderful thing about plasmids is that they can be easily engineered and can introduce foreign DNA into cells through electroporation or other methods. Many plasmids are designed so scientists can insert genes they want to be expressed in organisms. One way to do this is through restriction sites. Restriction enzymes recognize these sites and cut out the present gene like molecular scissors. Then, a different gene can be inserted into the site. Often, these restriction sites are located in what's called a multiple cloning site, a short segment of DNA that contains several restriction sites. This adds flexibility to the cloning process. How is the inserted gene expressed? A promoter site, which is upstream of the inserted gene on the plasmid, acts as a green light that allows gene transcription. RNA polymerase binds to the promoter, moving along the strand. As it moves along the strand, it creates a new strand of mRNA expressing the gene. The plasmid cloning process is very versatile, so there's a whole array of genes that scientists can introduce into the cell. For example, if you wanted to track a specific species of bacteria in a population, you could insert a GFP-expressing gene into a plasmid and transform it into your bacterium. Your cells will fluoresce, making them easier to find. Or if you're trying to study the effect of a specific protein on a phenotype, you could insert the gene of interest into the plasmid, move this into a cell, and look for changes in the cell. We hope you enjoyed this video in our Plasmids 101 series. To catch our newest videos,